So my name is Furupero Tambani, and I will be facilitating today's parallel session on showcasing your science. I have Ketelo Dinala, who is running the tech. So she'll be the one sharing everything that needs to be shared in the session. And Osisipo Sochangane will be her backup. So just in case, I see we lost Usi. Just in case one of us drops off or something like that. So thank you everyone for joining us, and we hope you will enjoy this session and just getting to see what other people are doing with their science, what research is going on, because we don't get much platform to share our science. So I hope we will enjoy each other's work today. To start or to open, I will just introduce each speaker as they are about to speak, instead of just introducing everyone all at once. So to get us started will be Marinda Koekemoer. She is the Deputy Director of Agric Sciences at SANBI, and her presentation is on group projects, a career lifeline. She will describe, or she will just let us know what she means by that. I'm sure people have questions in their minds, trying to piece together what to expect, but she will explain everything. And Marinda will share her screen. So Marinda, please go ahead and start sharing your screen. And then you can take it over from me from here.
So, good morning, everybody. Hester and Pieter challenged me to do a presentation today, and they actually asked me to speak on the work that we're doing on the plant families project. But when I started the presentation, I thought maybe I can share a few lessons. Basically, my journey at SANBI, working in group projects of several kinds. So I started working at the organization in 1987. So now it's 35 years later, and most of you would see me as a fossil. But I've been at the organization for a long time, but within this journey, I see my career basically on three levels. I started off as a technician, then went into management, being the curator of the herbarium, and currently I'm in a research position. But I think throughout one's career, one gets all sorts of opportunities, and you get opportunities to work with fantastic, great people, with great visionaries, with people that you can learn a lot from. Over such a period, you also deal with changing technologies all the time. And in this process, you build a lot of knowledge and skills until you at a level where you start initiating your own projects. And this is basically what I'd like to focus on is the process, what you learn from it, and the kind of exposures that you get throughout a long career with the same organization. So most of you may think it's unimaginable to stay with the organization for 35 years, but you'll see in in this presentation, there's been a lot of changes. There's been a kind of like many opportunities of reinventing yourself, and I think that's the crux of staying with the organization for a long time. So I've been involved in several projects throughout my career, and these are just some of them, and I'll touch on some briefly in the rest of the presentation. So the first thing I got involved in was the grasses of Southern Africa. So soon after I joined, then we were still the Botanical Research Institute. My boss was Dr. Beth Gibbs Russell. And on the first day, I had a very big shock to learn that she could not understand a word of Afrikaans. So here was this youngster Afrikaans girl coming into the organization, and suddenly faced with everything is in English, all the reports, all the interactions, the meetings, everything is in English. So I had to pick up a lot of English very quickly. So, and in those days was like really primitive days. It was before everyone had their own computers. It was before internet and Google and all of that existed. So getting involved in a project like this, working on the grasses of Southern Africa, was already a daunting task, because grasses are seen to be quite specialized, and most people don't want to learn about grasses too much. But this book, and working with Dr. Beth Gibbs Russell, was actually a fantastic experience and a fantastic first exposure for me as a youngster, working with a person like Beth Gibbs Russell. It's the person who started off doing checklists for Southern Africa, and we've seen those checklists grown until today. It forms the backbone of our Brahms database. So she was really a visionary that helped us through lots of things. So those days we had the pricey data already existed in a mainframe computer that sat in the city center of Pretoria, and we had a few terminals. There's little black screens with green letters on them, and we had a few terminals scattered throughout the building. But this book as such was the first in many respects. It was the first family that was treated for Southern Africa. It was also the first computer-generated book. So we worked on programs like Dawson Delta and IntKey and things like that. WordStar was our state-of-the-art word processing file software. And in those days, it was great. Beth had a computer in her office, so she did a lot of her work on a typewriter still. So one of my big tasks was doing the spikelet photographs. As a technician, I was saddled with all sorts of things. And the whole process of doing the spikelet photographs was quite daunting, because we had a fairly primitive type of microscope with a camera. We still worked in black and white film, so you couldn't immediately see what the results are. I had to do quite a lot of experimenting to find the right lighting, the right background, the right, which you had to get the spikelet in focus, but you had to get the background out of focus. And eventually I found that mounting the spikelet on a tip of a pin and pressing that pin through a very fine steel sandpaper, and then mounting it horizontally onto the microscope platform, I got the best result. But it was quite tricky to balance the spikelets on the tip of the pin. But anyway, it was exciting, and it was great way to start off with. The page that you see on the left-hand side of the screen is a page from the book. And in those days, there were no typesetters or anything like that. We used to prepare bromides of all the text and the maps and the illustrations and everything. But then we would sit and cut out these little bits of blocks of text and manually lay it out on a print-proof page. So it was a very time-consuming process. We learned lots of new skills. You had to make very sure that you align the text and you get everything laid out properly onto the page. So that was my first big project. I then worked with John and Heidi Anderson, and they famous paleobotanists. They needed a volunteer to work after hours and help them basically do the same page layouts for their books. I didn't work with them a long time, but what I learned in this process is to work with scientists that's really, really passionate about their work. You know, their attention to detail, the meticulous way in which they handled everything is really was a good lesson for me. After that came the Sabinet project. And I see I'm very quickly running out of time here. So the Sabinet project was a huge project in which we became involved with basically, at that stage I was curator of the barium, so it was a training and capacity building project led by Chris Willis, lots of reports. We got involved in the barium techniques, which is like a small version of the NSCF manual. Lots of training courses, identification courses, and that. Again, expansion on checklists, also through the Sabinet series. And of course, field trips that we loved. So each of the 10 countries got a new vehicle, and we all trekked up to Malawi in our brand new vehicles. So along came 4x4 driving experience and dealing with huge field trips organized in a military type of fashion by Chris. But it's a lot of exposure to new environments with different cultures, different personalities, and all in all, wonderful experience, learning processes. And this photograph is one of Chris's famous photographs. It's the results of the Nika field trip in Malawi, and then the publication that came from that. After that came the API projects, the African Plants Initiative, which is basically a project scanning type specimens, illustrations, and artwork. It was a very intense project with lots of staff members for a short period. And the results are stored in JSTOR. And it's because of the API project that you can now access type specimens and artwork through JSTOR of most of the types of South African plants. The project was later expanded into Europe and also into Brazil. So JSTOR has grown in this process. But through this also came the learning how to deal with really big projects like this, new technology, scanning of the specimens, photographing of specimens, all sorts of new things that we learned in that process. And this is just, yeah, every picture tells a story. So, but I don't think I've time to expand on that one now. The other type of project one gets involved in is research. Now, it's often a individual project, and it's often a very lonely process, but you learn all sorts of new techniques, writing project proposals, funding applications, publications, learning about microscopes, collaboration, all sorts of things. So the last project that I got involved with was the Plant Families Project. And so Stelitzia 31 was the guide to the plant families. It's our first publication. It dealt with the 52 large families. And now Stelitzia, and we think it's 46, but it's maybe another number, but it will be published next year. It deals with all the flowering plant families, 4,500 images, 2,200 images named to species level. And it's going to be a huge doorstop of a book, over 700 pages. But again, working with a winning team with two fantastic colleagues. We encourage each other, we challenge each other, and it's a learning process all the way for all of us. And each one brings their own skills to a project. As a last thought, the moral of the story is, well, maybe I'll just skip to the last point and say, you know, NSCF is a project like the ones I've illustrated before, and please make the most of it, because it's a wonderful learning experience. It's a process that challenges you and each one of you, and have fun and enjoy the process. And I'd like to leave you with this thought. If you find your passion in life, hang on to it. Learn and develop as much as possible to grow to your full potential. You are shaping your future, no one else. Have fun. Thank you all.
Thank you, Marinda. What a career. Wow. Like I said on the chat, it just feels like a whole new world as you're explaining it. Before computers, before Google, we have it easy now, but I can just imagine how difficult it was before all this, having to look for books and always being in libraries, learning as much as possible from colleagues and mentors. So thank you for sharing that. I feel like it was such a good way of starting the session, just having a word from such a beautiful career that transgressed, that grew from then, when you couldn't even really communicate in English and all you knew was Afrikaans, and having to learn English on the job. I can just imagine. So thank you so much for sharing your story. It was quite empowering and inspiring. And I hope that even people in the room found inspiration from it, and just your words at the end to just make the most of what we have. Make the most of the NSCF. And as part of the NSCF Hub team, we are here, right? We are here to help, to assist, to give to do whatever we can, as much as we can. So thank you so much, Marinda. Right, so now we will move on to the next presentation. But before we do that, just to let you know that you can paste your questions on the chat as people are presenting, just so that you don't forget. Just in case you don't have a notebook, you can put them on the chat. We will keep an eye on the chat. And at the end of all the presentations, we are going to have a session for Q&A, so you can ask your questions. Right, the next speaker is Pieter Besta. He is also from SANBI. He's a control scientist. He is not in the room with us, but he did send us his presentation. So Ketelo is going to play that. His presentation title is the Waterberg Mountain Complex Biodiversity Project, Progress in Spatial Planning, the Sampling of Plant Specimens. And he says that he will give a short overview of the project, showcasing the plant collection aspects thereof. So let us watch or listen to Pieter's presentation.
Good morning, everybody. Today I will showcase the Waterberg Mountain Complex Biodiversity Project, a project that I am currently involved in. This presentation is based on various presentations given by Prof. Nigel Barker from the University of Pretoria and the August newsletter compiled by Marlies Greiling. The project is funded through the Department of Science and Technology and is administered through the NRF by the Foundational Biodiversity Information Program run at SANBI. This project brings together various institutions countrywide, as well as stakeholders from the Waterberg itself. The project has been showcased in various newsletters, like the SANBI Gazette, which featured various contributions on the project. At the end of August, the first newsletter of the project was published to show the activities of various teams working on the project. It was compiled by the project coordinator and manager, Marlies Greiling. The team consists of a large group of scientists, lecturers, students and interns of a wide range of organizations and universities, investigating 7 broad organism groups and also looking at climate change and remote sensing, as well as community engagement. The project was initiated by Prof. Nigel Barker that secured the funding through a successful FBIP project proposal. The project actively started in April 2021. The funding is for 3 years and it comes to an end in March 2024. Marlies Greiling is the project manager. She is a local resident of the Waterberg, and her main function is to liaise between scientists needing to visit their study sites and the landowners who form part of the project. Collecting field data could only happen once permits for collecting material of the various taxonomic groups was approved by LEDET, who is the issuing authority for permits in the Limpopo province. To date, surveys have been done in 12 reserves that fall in the 10 priority grids within the Waterberg. Teams from all taxonomic groups collected baseline data and are currently planning more intensive data collecting trips. So why is this project important? The Waterberg Mountain Complex is a discrete geological entity that falls within the declared UNESCO Waterberg Biosphere region. It is home to 3 provincial nature reserves and 1 national park. Proposed coal mines and related infrastructure projects on the northern and southern borders of the area represent a major potential environmental conflict and will impact the geological integrity of the Waterberg mountain complex. This region is of special conservation concern, and this is the first ever comprehensive and structured biodiversity survey in the region. The objectives of the project is to liaise and collaborate with Waterberg stakeholders and organizations involved in biodiversity conservation and management of environmental resources, amongst others. To establish a database of existing grey literature, to undertake multi-layer baseline biodiversity surveys for a range of plant and animal groups, to assess species and habitats of special concern in terms of their continued conservation and management, to undertake DNA barcoding on samples collected and augment various databases on the national biodiversity of these data, to provide and make freely available all data and information to all stakeholders so that regional, provincial and private conservation and management plans can be improved and adapted, to have community engagement through education and citizen science, and additionally to have floristic and faunistic analysis of endemism and biogeographical affinities at the end of the project. The study area comprises the mountainous areas broadly between Lephalale and Marken in the north and Tabazimbi and Bela Bela in the south, with most of the priority grids in the drier northern parts of the Waterberg mountain complex. Priority areas to survey was based on cumulative QDS diversity rankings based on known plant, reptile, amphibian, beetle, bat and mammal data. The approach to surveying and assessing the region's biodiversity will adopt a two-pronged approach. The first year consists of numerous intensive surveys across the 10 least known QDSs. The following 2 years will focus more on selected areas or reserves considered to be of biodiversity and strategic value, and to do DNA barcoding of samples of selected taxa. Surveys in the second and third years will comprise of seasonal sampling across multiple years in such a way that it may be used for long-term monitoring. The focus areas for research is mainly on state-owned nature reserves and private game reserves or farms. In addition, in the last year, surveys will be conducted in areas of habitats noted in previous periods as of being of special concern or interest. The plant teams started collection in December 2021. To date, a total of 1,853 samples and their duplicates were collected on various reserves and their surroundings, covering at least 9 quarter-degree grid squares. Plant specimens collected throughout the project are returned to the various herbaria of participating institutes for processing and identification. These are the Swikert Herbarium at the University of Pretoria, the Larry Leach Herbarium at the University of Limpopo, and the National Herbarium in Pretoria. The focus of the mammal team is mainly on small mammals, including bats and rodents. Plot surveys utilized is non-invasive passive sampling with the use of camera traps and audio moths that sample and store the ultrasonic calls of bats. Camera traps are used to get overall distribution records, and the audio moths recordings are analyzed to identify the bat species. To date, 44 mammal species have been identified, including bats from 3 families. Aquatic surveys include mainly fish and aquatic macroinvertebrates. To date, 18 native freshwater fish species have been collected and identified, with 2 new records for the Welgevonden Private Nature Reserve. The aquatic macroinvertebrates group sampled 23 sites in a week-long recce trip, identifying 11 broad family groups. Aquatic macroinvertebrates in wetland areas were surveyed recently by members from the Rhodes University and SANBI interns. Samples are currently being processed in the lab to extract DNA, which will be stored in the SANBI biobank.
It seems the video has
The
stopped.
histology team collected 127 voucher specimens and DNA samples representing 8 amphibian and 28 reptile species. The endemic Waterberg dragon lizard and Waterberg flat lizard were sampled at all 5 properties they visited during their recce trip. As part of the project's community engagement, Prof. Barker introduced children of the Waterberg Academy to iNaturalist. In the workshop he held with them, they were shown how to upload images of biodiversity taken with their cell phones to the Waterberg's dedicated iNaturalist project. To date, many bioblitzes have been done in as many as possible priority areas. In the next 2 years, the main research sites will be established with ongoing collections from other areas. Contact persons for the project is the head and manager. You can also follow progress on the Facebook page of the Waterberg Research Support Center. Thank you.
Thanks to Pieter and such amazing work. I'm sure our SANBI colleagues here are quite proud of such work. It's so important to have a record of what we have in our biodiversity. So all this work is very important. We will now move on to our next speaker. So we have Janne Delport. Janne is a PhD student in melisopalinology. I had to practice this at UCT, and she will be presenting live, and she will share her own slides. So the title of Janne's presentation is "The Cape Captured in Gold: A Pollen Perspective." The first time I heard about her research was when we were doing the video that we all watched yesterday in the forum, and I was just amazed by the kind of work that she does. So I'm interested, I'm quite excited to hear what she has to say about that. And to describe her talk, she said melisopalinology is the study of pollen and spores found in honeys. She will elaborate on the insights to be gained from recording the pollen composition of honeys and how they enhance national benchmarks for forensic food authenticity testing. Quite interesting. Are you ready, Annie?
I am. Thank you very much. Can everyone hear me clearly?
Yes.
Perfect.
I see you are making use of our theme, and it's like a perfect fit.
Yes, I also thought so, and I was like, I have to. I can't do the presentation and not have this.
Yeah.
Okay. Can everyone see my screen?
Yes, we can see it.
Perfect. Cool. Good morning, everyone. My name is Janne Delport, as Fulu has introduced me, and I'm a PhD student from the University of Cape Town, where I'm currently busy studying honeys collected from the Cape Floristic region, and I'm specifically interested in how we can use melisopalinology to characterize the honeys and develop benchmarks by which we can test the authenticity of honeys in the future. But before I get into that in too much detail, I'd like to introduce everyone to palynology first. So palynology is the study of pollen grains and spores, And the success behind palynology is the morphological diversity that we can see from different taxa and their pollen, especially when we consider size and shape and ornamentation. The diversity is quite intense, and it has been a pretty useful tool for a variety of fields. So the consistency of characters between taxa varies from group to group. In some groups, such as the Asteraceae, the pollen morphology is quite consistent for the family. So in general, they are known to have quite spiny pollen, whereas in other groups, such as the Acanthaceae, they are notorious for having what you could say quite unique pollen, with some of their pollen grains looking like little spaceships or tic-tacs. So the level of differentiation using pollen morphology differs from taxa to taxa, and in some cases we can only identify to, for example, a family level, but with other taxa and other groups we are able to identify them to genus, and in some cases even to a species level. Palynology has been incorporated into quite a variety of fields. In medicine, it is used to identify and track allergenic taxa throughout the seasons, and they use this to be able to let people know when they should expect some seriously allergenic time periods, especially for people who are very heavily affected with, for example, hay fever and that sort of thing. In forensic science, it can be used to determine or to infer the origin of products. For example, if there's contraband, they can use pollen to infer where the products are from. They can also use the pollen morphology to link suspected criminals with specific crimes as well. In the field that I'm interested in, which is melissopalynology, the study of pollen in honey, you can use it mostly to get an idea of the diversity of taxa that the bees are foraging from, and it also helps us establish what type of honey is being sold commercially, and also a general idea of where the honey is from. So melissopalynological studies can be divided into two sections, mostly focusing on the botanical origin, but then also the geographical origin. The botanical origin is used to determine what type of honey it is, and the honeys can be divided into two groups, namely monofloral honeys and multifloral honeys. So monofloral honeys are products where the predominant pollen composition comes from one source, and some denominations that you may be familiar with if you buy products from the supermarket or that sort of thing. The most commonly known monofloral honeys are citrus, or for example, eucalyptus honey. Multifloral honeys are considered as such because there isn't one particular dominant pollen source, therefore they have a more generalized or broader composition, and the denominations that these honeys are sold under generally include things such as wild blossom honey or wildflower honey. So the botanical origin is mostly used to determine the appropriate denomination of the honey and basically giving us a better understanding of the type of honey that it is. The geographical origin, as the name implies, is a way for us to determine where the honey was harvested from, whether it would be at a national level to determine which country the honey is from, or in some cases it can even be linked to a specific region. So we are able to tell that some honeys have been produced from a very specific region in a country, for example, maybe belonging to a particular province, or what I'd like to see in the case of South Africa is whether we can associate it with the Cape Floristic region itself. So before we started analyzing all of the honeys, we wanted to confirm where we should focus our efforts first in terms of research, because you don't want to reinvent the wheel necessarily. So I did a review on melissopalynological studies across Africa, and what we did is we had a look at the outcomes of these studies to determine the most efficient way to do honey research so that it directly contributes to the honey industry. What we found is that we can develop there's a specific framework by which you can develop your research, with the main focus of the studies being recording the pollen spectra of honeys and then subsequently profiling the honeys. And if you do this, you are able to develop the benchmarks that are necessary for, in the first place, to determine whether the botanical and geographical origin of the honey is in fact true, but also can be used to verify whether the products that are being sold on the market are truly from South Africa, if that is how they are labeled, and whether the denomination that they're being sold under is also a true reflection of the actual product. So that is how the baseline research of recording the pollen spectra and the profiling can feed into assisting the honey industry, which overall supports the beekeeping industry because it reduces the prevalence of fraudulent products in the market. What we found that was quite shocking is that for South Africa, up to date, there's only been one study done on the pollen composition of honeys. And what we found is that it doesn't fit in with the baseline framework research that we saw earlier, which is the recording of the pollen spectra or either the profiling of the honeys. So if we consider that, it means that we have no way actually of determining the botanical or geographical origin of the honeys, and we also have no way of verifying other products against this record that we have already. So in general, there isn't any pollen composition studies available for us to do any benchmark measurements of our honeys, which means that the industry is quite at a disadvantage because we just don't have the support, we don't have the academic support to give to the industry itself. Thus, for my thesis, we decided that we would focus on the two baseline studies. So I will be focusing on honeys from the Cape Floristic region, and we'll be looking at recording the pollen spectra in the honeys. So getting a better idea of which taxa the bees are interested in, what their prevalence is, whether we have any unique monofloral honeys from the region, and assigning the honeys more accurate denominations that they can be sold under. I also wanted to investigate the geographical origin of the honeys, mostly to see whether we can differentiate the Cape Floristic honeys from products produced in other countries, products produced in other provinces, and to some extent, I also wanted to see whether differentiation is possible within the region, specifically considering the major vegetation zones that occur in the region, for example, fynbos and the succulent karoo and that sort of thing, considering the diversity that we're sitting in at the moment. So for my thesis, I collected 78 honey samples and recorded the pollen spectrum for all of them, of which 74 of them have been harvested from the Cape Floristic region itself. What I found in total is that there were 18 monofloral honeys recorded, and some of the denominations that we found were suitable for them are, for example, citrus. We had some canola honey, and we had some echium honey as well. To be multifloral, and in total of the 78 honeys that we studied, they recorded 91 pollen types, which roughly translates to about 91 different taxa that the bees foraged from. And this diversity of taxa has set an incredible basis for us to do the geographical differentiation because, as I said, there's just so much to work with. So we've got a good opportunity to see to what extent and to what scale we can differentiate the honeys. What we found is that if you use a non-targeted approach, so specifically just looking at the pollen composition as it is, we are able to differentiate honeys produced within the region from honeys produced elsewhere. So the non-CFR honey samples in this graph are honeys from Limpopo. There's a honey from the Northern Cape, and then some honeys from Zambia as well. So when you're looking just at the pollen composition itself, we are able to differentiate them at a national but also provincial scale. But when you try to consider the differentiation within the region, it doesn't seem to work as efficiently as we expected with this particular method. We then considered using a indicator species method instead, so that we can see if we associate the taxa of the honeys with their corresponding vegetation zones, whether that would allow us to differentiate the honeys more efficiently. And we found that it was quite successful. We are now able to differentiate honeys produced in the region according to 4 major vegetation zones, that being the fynbos, renosterveld, strandveld, and succulent Karoo. So what this means for the honeys produced in the region is that we are now actually able to assign proper commercial denominations to them. So we are now able to suggest to beekeepers that if they want to sell honeys under a specific name, they are able to do so based on the pollen composition. They can sell their product as a locally produced authentic fynbos honey or Karoo honey. This is important because it means that we can add more value to the local products, even if they're not necessarily considered monofloral honeys. They would still have sought afterness due to it being associated with the Cape Floristic region. This differentiation has also helped us to establish the generalized pollen compositions that we wanted, as I noted in the framework. So now we are able to verify the origin of future honeys and future commercial honeys as well, to establish whether the name that they're being sold under is in fact true, and whether the products that we have that are being sold here are actually from the Cape Floristic region and are authentic in that perspective. What I'd like to also emphasize is that all of this research that I've presented to you up to now has only been possible because of the accessibility and the availability of pollen identification tools. So one of the major tools that we use as pollenologists to identify pollen is reference collections. So during my thesis, I wanted to expand on the pollen morphology knowledge that we have for Melitophilus taxa in the Cape region, in the Cape Floristic region. And for that, I relied on specimens from the Bolus herbarium, so at the University of Cape Town, so that I can collect pollen from. So I relied on specimens from the Bolus herbarium at the University of Cape Town to collect my pollen from. This has been beneficial for a variety of reasons, one of which being I have a guarantee that the taxa that I collected the pollen from have been identified by experts. So it is almost guaranteed that the taxa have been identified accurately. And we can link the pollen reference slides with the voucher number of the specimens as well. So in the case where there are any taxonomic changes in the future, when the changes are made on the actual specimens at the herbarium, that can be corresponded then with the appropriate slide as well. So by using herbarium specimens, I've been able to ensure that the quality of the slides I've been producing are very high, which in so would allow for higher quality research to be done in the future. The slides are currently being stored at the University of Cape Town in the plant conservation unit. And what I've done as well is that I have digitized these slides, because that is one of the bottlenecks when it comes to these reference collections, is that various institutions across South Africa do have collections, but they're not necessarily digitally available. So they are only as accessible as they are locally, because they are limited very much to the institutions themselves. There are quite a variety of ways that you can digitize the collections. One of the databases that you can contribute pollen images and photos to is the Global Pollen Project. It is a free platform that is available for anyone to use, obviously, and you can upload your pollen photos there, and basically it's just an online database where they're trying to make pollen identifications more accessible. And at this point, it is an international project, and contributions are freely available. So you can if you are confident in your identifications and that sort of thing, or if you have a research article with pollen morphology in it, you can easily contribute your photos there and assign the respective DOI and the article information as well, if people wanted to follow up. Otherwise, other options include atlases. South Africa does have 3 volumes of a South African pollen atlas. However, this is also not digitally available yet for everyone to use. So there's still a lot of digitization that needs to happen in terms of our pollen resources in South Africa. So as much as that framework showed, or the review showed, that there's still a lot of melissopollinological research that could be done, there should also be emphasis placed on pollen research and pollen digitization, because not only will this help me with my field, but it will also contribute to other research fields such as paleobotany, paleopollinology, medicine, forensics, etc. So yeah, that's all that I wanted to talk about. Thank you very much for listening, and I hope that you guys have gotten as excited as I am about pollen.
Thank you, Yane. It's quite interesting work. I think from now on, I'm just gonna look at the bottle of honey that I buy. I'm gonna read instead of just buy, and look at the pollen content if they put it there. Thank you. We are running a bit behind time. So I'm just gonna ask Bernard to go next, and then that will mean that we have 2 videos to play after him. I just want him to have time to present because he is here. Bernard, will you share your screen?
I'll try to make this quick.
Thank you, Bernard. So I'll just introduce you. So Dr. Bernard Zipfel is from the Wits ESI, and he is the curator of fossil and rock collections there. His presentation is an overview of coprolite collections at the University of the Witwatersrand, and do they matter? He's going to let us know if they actually matter. So I'll take it over to you, Bernard.
Hi, everyone. Just in the interests of full disclosure, I'm not a coprolite expert. I'm not a palynologist, and I'm actually a functional morphologist in hominins. But because someone suggested that some of my research I do is crappy, I decided to do something on coprolites, which, for those who don't know, is in fact fossilized feces. So I'm doing this more tongue-in-cheek and for fun. So there's a number of colleagues that have collaborated with me to essentially find out whether our coprolite collections are important or not. Actually, I could answer the question and say, yes, they are, and we could just leave it at that, or I could maybe just tell you why. Maybe I'll just tell you why. So we have Chandelier Montgomery from Wits, who's doing her PhD on coprolites. Frank Neumann, who's a palynologist, who's at the University of the Northwest. Jonas Chouanier, who's a vertebrate paleontologist. Louis Scott, who's also a palynologist from University of Free State, and John Hancock, who's a vertebrate paleontologist part-time at Wits, who started looking at, sorry, could you, I don't know how to advance the slide. There, thank you very much. So to tell you what fossil poo, for want of a better word, looks like, that's what it looks like. Some of it actually looks like feces, and some of it is less identifiable. And I found this interesting little thing on the internet, actually Facebook. It's called the Pooseum, and it's #1 for fossilized #2. But it has got a nice little diagram here telling you how to identify a coprolite, because that, I think, is where the problem lies in the fossil record, that often we don't recognize these things. Firstly, by the shape, does it actually look like poop? Does it have burrows? the location? Is it found within the vicinity of other fossilized materials, skeletal material? And does it have any inclusions, bits of bones, scales, fins, teeth, that sort of thing? And then in the chemistry, does it reveal any interesting sort of dietary things? Next slide, please. One of the colleagues you saw on the previous picture was John Hancock's, who had for years collected lots and lots of fossil poop, literally thousands of them. And one of the things he asked me when I said, you aren't cataloging your material properly, he says, well, I've got 30,000 coprolites here. Am I supposed to catalog every one of them? I said, no, just dump them in a box. But we've had to backtrack on that because we're actually not treating this fossil material fairly. And we decided to embark on just a very short little study on trying to find out how much of this fossil poop do we have in the collections. Now, our collections span from the Permian right through to the Holocene. So it's very different environments, very different fossilized material. But we looked at 21 collections we have, ranging from firstly the Karoo vertebrates, which are dinosaurs and therapsids, and then the trace fossils, which theoretically should contain these coprolites, right through to mostly Pleistocene sites from the cradle. And we thought, let's have a look at how much of this has ever been published. To our surprise, there are only 6 papers published directly on coprolites from our collections, and these came from 4 distinct localities. So in the bottom left, we've got from the most recent, that's human fossilized feces. I never thought I'd put this in a presentation, bizarre, but that's human poo, which is about 1,100 years old, published by Rian Rifkin and colleagues. And there they found some very interesting dietary issues, which showed that the Bantu speakers, Iron Age people of that time, were pastoralists, hunter-gatherers, and agriculturists, a mixture of those traditions. The calcified latrine from Gladysvale, headed by Lee Berger, showed that the hyenas, brown hyenas of the time of around 250,000 years ago, were doing exactly the same kind of latrine culture as what we see today. Lucinda Backwell took that same material and actually found human hair in it, which showed that these hyenas were in fact chomping on human tissue. And there we've got human hair aged at, let me just remind myself, around 200,000 years ago, which is the oldest human hair ever found, if indeed that is true. Then Marion Bamford looked at coprolites coming from the famous Malacca site where Australopithecus sediba was found, and they looked at the palynology. There was some pollen, there were some wood fragments from the area, and also phytoliths. This is silicified micro elements of the poo. So Janae, if you want to move from golden honey through to poo, you're welcome to do so. And then we had on the bottom right, you see a picture of Adam Yates doing work on some Karoo. This is Triassic, early Triassic coprolites. They contained bivalves that hadn't been seen before. So we then looked at the presence of coprolites in the Historic Evolutionary Studies Institute fossil collections, and we found 10 of those 21 sites had semi-catalogued specimens. Some were not catalogued but put in separate boxes, but we did find them. And strangely enough, from the Karoo vertebrates of 9,000 specimens, there were only 40 that were listed. And the question was, why did we only find 40 coprolites that are in the collections noted, where John Hancock's found 30,000 that he's put in boxes? The answer is probably people don't recognize them and just chuck the bloody things out. So, yeah, on the others, Gladysvale, Swartkrans, Stagfontein, Kromdraai, Cooper's D, they've all got coprolites. Unfortunately, a little boring because they all seem to be from carnivores, either unknown carnivores, but most of them identified to probably be hyenas. Various hyenas, possibly extinct hyenas, but a lot of them looking similar to the extant ones, which is a very common thing in southern Africa. So here's just some pictures of coprolites of potential hyena origin from Plover's Lake, Gauteng. We did also notice that some of the shapes and sizes of these things looked a little different from site to site. Whereas, for example, from Swartkrans, some of these coprolites looked, for example, like anatomical elements. Because remember, these things are fossilized. For example, like a head of a femur. And in fact, may have been misdiagnosed originally. And if one looks more closely, it has the shape of the coprolite. Here's some early Triassic coprolites from the Driefontein farm, those many thousands that John has been finding. And one of those from the early Triassic was CT scanned, and we found a therapsid lower limb in it. And that is currently under study. We're going to synchrotron that at the ESRF in France and get a higher resolution. We're not particularly interested in the coprolite, but we're interested in this particular vertebrate that's in it, and it's preserved in a way that we wouldn't actually find outside of a coprolite. So that's the value of this material. There you can see some Triassic coprolites from the Karoo vertebrate specimens. There's some more coprolites from the Permian. Those are even older. Those are closer to 300 million years old. And Chandelay, our student who's doing her PhD in this, has initially CT scanned many of these specimens, and you can see the beautiful material that can be extracted from them. There are scales, cranial elements, there are beetles, fragmentary insect remains, there are bivalves, there's lungfish, and of course pollen as well. These will also be synchrotroned for a higher resolution. What Chandelay is also interested in is that we look at the actual coprolite and to develop morphotypes that can identify the species. For extant material, and of course into the Pleistocene, we can do that because we have comparable modern material. For dinosaurs and therapsids, not so much. So just an interesting thing, when someone says, what's that? And they pick it up and they look at it, I say, well, that is coprolite. They say, oh, that's very nice. What is it? And I say, well, it's fossilized poo. They immediately hand it back to me and ask where the bathroom is if they could wash their hands. So it shows how people respond to these things. So in conclusion, every paleontological repository should carry out an audit on the coprolite collections, ensure that they are documented for future use, communicate to geologists and paleontologists what these things look like and that they're common. Every site has them. We must find them, pull them out and treat them like proper heritage material. The coprolite literature, the poo literature, commonly studies a few unusual specimens, but other than that, we don't see that much. The science is growing and there are scientists that are beginning to specialize in this field. More documentation of the distribution of coprolite morphotypes and ichnotaxa will allow broader patterns to be recognized. The study underway on the aforementioned Driefontein collection will go some way to achieve this. We've actually developed a separate catalog for them. And coprolites must be cataloged and the storage location formally documented. So that is it from me. So now you know what a coprolite is, and if anyone is interested in working on these things, we have loads of them. Thank you very much.
Oh my God, Bernard. I have to say, all I can say is you made my day.
I'm so glad, Fulu.
Thank you so much. I see Kirsten is also saying thanks for an entertaining talk. You brought life to the coprolite, so thank you so much. Time is not on our side, so let's go on to our next talk. It's a recording. It's Dr. Sibusisiwe Ngulana. She is the collections manager at Bay World, and she is talking about the taxonomy of bottlenose and common dolphins in South Africa. How many types of these dolphins are there on our coastline? Ketelo, please play a video for us.
Greetings, everyone. My name is Busisiwe Ngulana. I'm a collections manager at Bayworld. I did my PhD with Dr. Agric Meyer, who is the curator of marine mammals here at Bayworld. So my talk today is about assessing the taxonomy of bottlenose and common dolphins along our South African coastline, meaning how do we assess the number of species each of these genera of dolphins has. I would also like to thank the NSCF for providing opportunities like these so that one is able to present their work. So dolphins, porpoises, and whales fall under the infraorder Cetacea and are thus referred to as cetaceans. There are two groups, baleen whales on my left and toothed whales on my right. Toothed whales are referred to as odontocetes. They are the most diverse group of aquatic mammals with around 93 living species accepted. However, there are ongoing uncertainties regarding some types or species, and they require regular revisions. Bottlenose dolphins are scientifically known as the genus Tursiops, and common dolphins, which are scientifically known as the genus Delphinus, are amongst the most widespread and well-studied dolphins, but they are still subject to considerable uncertainty regarding the number of species or types in each group, and also their distribution. For bottlenose dolphins, it was thought that there was one species until a second one was described. So the description of these two species was done by Graham Ross, who also was a marine mammal curator at Bayworld at the time. And the first original species was the common bottlenose dolphin on my left, which is distributed in tropical and warm temperate oceans globally. And in South Africa, this type of dolphin is distributed inshore and offshore in the west coast and offshore in the east coast. The second species, the Indo-Pacific bottlenose dolphin, scientifically known as Tursiops adancus, is distributed in South Africa from the Cape Agulhas to southern Japan and to the northern coast of Australia. It is an inshore species. With common dolphins, one species was recognized, the short-beaked common dolphin, also known as the Delphinus delphis, and in 1994 a second species was described off the North Pacific coast. This species was known as the long-beaked common dolphin, scientifically known as Delphinus capensis. After this study, all common dolphins along the South African coast were referred to as the long-beaked common dolphin, following a study that was done for the South African common dolphins, as they showed similarities to this second type in this group. However, in 2015, another study suggested that even though these dolphins had different characteristics, morphologically, genetically, they were still one species, and thus we were back to having one species in this group. And so the aim of this study was to assess the taxonomy of bottlenose and common dolphins, specifically populations off the coast of South Africa, using different lines of evidence, including DNA analysis, skull, and external morphology analysis. So I used skulls, teeth, and external measurements obtained at Bayweld and Iziko museums. These specimens were collected along the coast of South Africa. And I will explain why I used each of the methods. So I use skulls as they are frequently the most valuable and available skeletal part for morphological studies. They do show variation in size and shape depending on sex, geographic location, and can also be used to separate species or populations. They have also been used extensively in primates and seal studies. So how do we get data and analyze this information provided by these skulls? Well, you could use calipers and measure different parts of the skulls or scan them, but for this study I photographed the dorsal and ventral aspects of the skull. Then I landmarked the skulls. The red dots are the landmarks used for this study as they capture the size and shape of the skull. Programs such as PAST and Morpho-J were used for this analysis. Then I chose teeth for this study because we wanted to do genetic analysis on specimens that were used for skull morphology analysis. And for most of these, there are no tissue samples like skin or muscle to extract DNA from. And teeth are very good at preserving DNA better than other soft porous material. And so historical DNA analysis was used for this study. So historical DNA is DNA extracted from preserved organisms, parts thereof, or products made from biological material. So museum specimens such as teeth and bones are commonly used for this. Usually mitochondrial DNA is easier to amplify in DNA extracted from preserved organisms, and mitochondrial DNA is also useful in evolutionary and phylogenetic studies. Cetacean studies, including taxonomy and population genetics, have made use of historical DNA. So for analysis, getting DNA from teeth started with me bleaching them, grinding them to powder, removing calcium from them, then using the old but useful phenol chloroform extraction, DNA extraction method. Then the DNA was amplified, sequenced, and then eventually analyzed data on the different programs such as the J model test, Mega5, and Network. And lastly, I used external measurements and sometimes photos if they were available. These are taken before the animal was dissected. So external measurements and photos have also been used to describe variations within or among species, just like skulls. So they examine life histories, growth patterns, and also sexual dimorphism, which is the study where males and females of the same species are investigated for any differences. Body measurements and color patterns have also been extensively used in species descriptions, and there is always a need to describe external characteristics of an animal, as this would allow easy identification, especially in the wild. These were also analyzed to see if there would be any information that would provide characters suitable to separate different species. And what did the different analysis reveal for bottlenose dolphins? So all the three analyses revealed that there were indeed 2 species in this group of dolphins. External measurements showed that measurements such as the total length and the width of the flukes were some of the measurements that were important in separating the 2 species. From the skull morphology analysis, We could see that the smaller skulls were the Indo-Pacific bottlenose dolphins, while the bigger skulls belong to the common bottlenose dolphins. And what about the common dolphins? Well, none of the analysis showed significant differences between groups and supported that there was a single but highly variable species, both genetically and morphologically. The skull N420 in the black asterisk on the map, the last map on my right, was an outlier, and this skull looked a bit different compared to the other skulls, as its beak looked longer than the other skulls in the collection. So in conclusion, the pattern observed for bottlenose dolphin agrees with the most accepted taxonomy, that is 2 recognized species, the common bottlenose and the Indo-Pacific bottlenose. And the pattern displayed by common dolphins indicated a single highly variable species, the Delphinus delphis, rather than 2 distinct species. I must emphasize that these regional taxonomic revisions are very important, as they contribute to the global context regarding the taxonomic status of these dolphins, and also they are useful for conservation decision making regarding specific important populations reported. And finally, I would like to acknowledge these institutions and departments such as DST at the time of the study, NRF, and the State Serum Institute from Denmark for funding, Bayworld and KZN Shark Sport, and IZICO South African Museum for the specimens. Thank you so much for your attention.
Please play Pumlani's video for us. I guess Usi can. Okay, thank you.
Hello, it's me again, Pumlani Kwemi, the Albany Museum botanist. I'm just coming from the garden now of Observatory Museum, and now here in front of me, I've just picked up some of the wild plants that are in the Observatory Museum garden. One of them is the blackjack, which we are going to correctly identify in the herbarium. As you can see, this one is also another blackjack with all the flowers have got now seeds that are spine-like. And next to it, we've got Umgaleka, which is also another wild edible plant. Next to it, we've got Imbikikane, which we also called Chidopodium. Next to it, we've got Ihlaba. These we call Ihlaba. These are sometimes called dandelions, but the actual dandelion is this one. The one next to it is dandelion-like, and but because they perform almost the same functions used as wild edible plants, all of them, you can eat them as vegetables, you can cook them, or you can eat them raw. Very nutritional, as I've just mentioned. And secondly, they can also be used as medicinal. They do a lot of wonders. They've got lots of vitamins, mineral salts, proteins in them. They cover almost everything that our body needs. They cure a number of diseases that we are struggling with, especially this one, which we call the blackjack, has got all the antibacterial activities, antifungal activities, antimicrobial activities, and you can use it in order to cure a number of diseases. And as a result of that, if you eat them as food, you are not just eating them because they are nutritional, but you are also eating them because they've got a lot to offer for your body. For now, next to me, I've got a plant presser. I'm going to press all of them in this plant presser and take them to the herbarium for the correct identification before we can take them into the pot to cook for cooking, or we can eat them, so that we make sure that whoever is eating them is eating the right product. Other than that, let's wait until we get the results from the herbarium, because the herbarium has got all this information that we need to confirm the names that we have given to these, that they are the correct one and they are not closer. Because the danger here is that there are other species that are very close to this, which some of them, they belong to the same genus as this or to the same families, and they are deadly poisonous. If you make that wrong identification, you can consume something that can easily compromise your health. I don't want that. I want to be sure for all that is consumed is going to be consumed. For now, let's take the process of identification further until such time that I've got the correct names. Bye-bye.
Thank you, Dr. Klimi, for that. I know we don't have time. I'm so sorry that we don't have time for questions, and there was no question in the chat. I was keeping an eye, just comments and laughs and all that. So if there's a burning question, I can take it and then we go back to the main room. Any burning question? None? Okay, that makes me feel better, because it's not like I stopped people from asking. Thank you so much, everyone. I want to also thank every speaker who spoke. Thank you so much for all the information that you shared, for sharing your work. You can see how interesting it is. I was watching the chat, and yeah, that's how you can see that people are listening and they're enjoying the content. So thank you for making time, and thank you for attending the session. And I am sure you're leaving with something that you have learned from this, whether it's the laughs from Bernard's talk or the information, new learnings from Pumlani's talk, or Yane's about pollen and honey, or just the inspiring story from Marinda. I'm sure you learned something. So thank you. And we will now go back to the room. I'm sharing the link just to make it a bit easier for you. If you click on the link, it will take you back to the main room. See you back there. Thanks. Bye.